Our next speaker is Professor Dyann Wirth from the Harvard T.H. Sands School of Public Health, the Broad Institute, and also the Science of Eradication course.
Great. Thank you very much to the organizers. And perfect. All right. I'm pleased to be here to talk to you today. I'm very pleased to be here for the Alan McGill Symposium. He was a real leader and kind of visionary for many of us. And I'm really focusing today on talking about harnessing genomics for malaria eradication. And this is a topic that many of us, and there are many sessions during this meeting that focus on this. So I'm going to try to step back and think about this in a broader sense. As our previous speakers have shown, there's been great progress. We've done a great job of reducing both cases and burden, but things have slowed down a bit. And it's important for us to have new strategies and think about why things have slowed down and whether or not we can bring new thinking and new approaches to this problem. The WHO Malaria Global Technical Strategy focuses on that. And I just want to point out the third pillar of this, which is using surveillance, transforming surveillance into a core intervention. And that's really going to be the focus of what I'm going to talk about. And how can we improve surveillance, in particular using genomics? And so I thought it might be useful to think about this in a slightly historical perspective. If we look, and I wonder if I have a pointer, yes. If we look kind of comparing changing technologies, is malaria surveillance keeping pace? The telephone was actually developed around the same time the malaria parasite was discovered, and you can see the development of the telephone or telecommunications. And here's malaria development. Is it perhaps time for some new tools? And I would say perhaps genomics is there. Okay, so what am I talking about? Okay, so in the most conceptual sense, if we think about eradicating malaria, it's a process, right? It goes from lots of people with lots of parasites down to very few people with very few parasites, and eventually all people with no parasites. And so it is following that process where genomics can help us. So the current tools can count the number of people who are infected, but it can't tell what's underneath, what's actually going on. So people in areas of high transmission are often carrying many parasite genomes within them. As transmission reduces, this goes down, fewer parasites per person, parasite genomes per person, and finally to very few parasites that are actually present and circulating in the population. And I thought it would be useful, coming again to kind of looking at analogies, to think about how this might apply in the eradication setting. And to do that, I thought it would be useful for us to look at another disease that's in the process of eradication. And this is polio. I think many of us are familiar with polio. And here, genomics or genetic analysis has been a pillar of their surveillance and eradication program. And it plays many roles, in determining transmission patterns, surveillance quality, and program progress. I'm going to focus on two where I think there's really direct correlation of how, even today, we can use genomics to aid in the elimination progression. And then I'll move back to talking about what I think we need to do in malaria to transform malaria into polio with regard to the use of genomic data. So in the question of importation and export, every polio case today, the virus is sequenced and compared to an international database to determine origin and transmission chains. Well, we can do that today in malaria. I show here an example from our colleagues at the Chinese CDC where we've worked with them to look at in this case, a case of Plasmodium falciparum in a person who had no known travel history in a region of China that hadn't reported transmission in many years. By sequencing this, this parasite and then comparing it to the worldwide database that was developed by Malaria, and there was a session earlier today on that program of collecting sequences from around the world, we were able to very readily compare the unknown parasite to all of the parasites in the world, in this case using principal component analysis, and identified the parasite as being most closely related to African parasites, arguing that this is a case of importation. Similarly, in polio, one of the hallmarks of program progress is this reduction in genetic diversity of the in this case, the virus. And so we can actually take that same approach with Plasmodium. So if we look at a region of high genetic diversity, that is, people carrying many different parasite strains, sometimes singly or doubly, versus a population of reduced genetic diversity where all people or all patients carrying parasites are carrying exactly the same parasite as might happen in an outbreak. And so we can actually look at that directly, and I have an example for you of close to that. Oops. There. In Haiti, working with Malaria Zero and CDC, Sarah Volkman and colleagues have been able to look at the parasites in 3 regions of Haiti, where the number of parasites analyzed is very similar, but the composition is different. Where in one region, there's very strong relatedness of parasites. And here, using whole genome sequencing, you can see all of the parasites by principal component analysis are essentially identical to one another. This is clonal expansion of a single outbreak, whereas the other regions have more diverse parasite strains. So again, what we can see using the analogy of polio is we're already there in some ways with what we can do using genomics in malaria. The other thing, and this is a throwback to Al McGill, this is a slide actually from his presidential address when he was president of ASTMH, pointing out that resistance to both insecticides and microbial resistance, the parasite drug resistance, were the biggest challenges to malaria eradication. And we're already using, and have for years, this methodology of detecting resistance. And there are, I lost count at about 20 different sessions and hundreds of posters in this meeting on using molecular approaches to track drug resistance, to both discover and track drug resistance in various settings. So this is already ongoing work, and I think it's something we just need to kind of roll up and realize that we have this data, we should be using it in a broader way. And so here is just an example of using longitudinal samples from a particular region. In this case, this is work from Dan Neefsey's group at Harvard in the Broad, collaborating with groups in Southeast Asia to look at a particular population of parasites as artemisinin resistance was emerging, and find, in fact, that there are particular parasite strains or particular genes that are sweeping throughout the population, an early warning for the emergence of resistance, something that I think we'll see more and more usage. But in fact, in Africa, here in Senegal, at Czech Antidiop University in the PNLP, they've been tracking resistance to various antimalarials using molecular markers for 2 decades. And here's the very powerful antimalarial genetic group that has been working under the leadership of Jimdi from Mali to be able to look at Kelch13 potential emergence in Africa. So again, genomics is already being used in the eradication. But I think the next step, and the important step, and the place where I think we in the malaria community have the opportunity to set the standard for the field, is to actually integrate this information, which is primarily in research projects or in specialized silos, into the National Malaria Control Program and the analysis, and eventually into, sort of the vision is that we would use this in the same way that the polio community or the HIV community use their genomic databases. And I think this is a real opportunity, and we're poised to do that, in part because of the technological development. Every day there are advances in how quickly and how rapidly and how extensively we can get DNA sequenced. But the important thing is how we're going to use it to inform decision making. And I think that's the challenge that I see kind of for the next couple of years, is how you're going to take this vast amount of new big data and incorporate it in a way that is actually informative for those who are making decisions every single day on how their programs are progressing, how to distribute resources. And so I'm just going to give a very brief example from our own work in a collaboration with the National Malaria Control Program in Senegal and the Czech Anta Diop University, in this case the genomics part led by Professor Douda and Dyann. And this is an effort that's been ongoing now for a number of years, and there are several presentations at the meeting describing work in this collaboration. And so the idea really is that this is an iterative process. Well, there'll be a team that actually looks at the genomic data, generates the genomic data. Modeling will definitely come in, a very important part of this. There'll be technical validation, as is handled with standard routine data. There'll be political validation, that is getting the government, the decision makers, to actually decide that this is an important and valuable tool that they want to spend resources on. Then the actual use of the data in the decision making, and I expect this is going to be an iterative process. That is, polio didn't get it right at the beginning, we're not going to get it right at the beginning, but I think it's time for us to start. And so I'm just going to go very quickly through this because of time. Okay, so Senegal, just to remind you, is a country that has different burdens of disease or different transmission intensities, very low in the north, very high in the south. And so it allows us to be able to look at applications of these kinds of technologies in different transmission settings, but within one country and NMCP context. I remind you that what we're primarily measuring in this case is this diagram I showed you earlier, this reduction in the number of genomes in an infected person and the emergence of clonal parasites upon intervention. I'm first going to show you data where we have over a decade of data looking in Chess, Senegal, an area of moderate or intermediate transmission going to low transmission. And when interventions were introduced as we began this work in 2006, we saw an immediate increase in the clonal population and a reduction, therefore, in the overall genetic diversity of the population, consistent with there being a reduction in transmission. We've seen this reduction throughout, although there's been some variation, and modeling work we did with IDM earlier perhaps this indicates slight changes in transmission from year to year, but it needs much more work before we're able to say that. We also noticed that we saw parasites persisting year over year as interventions were introduced, as if this is a separate, perhaps, population of parasites that's there and remains and is part of a local transmission network. And I'll mention something about that in a moment. So here is some background data into which we can now begin to compare this to standard epidemiological data collected in these same sites. In the north, and Sarah Volk will be talking about this, this is a project in collaboration with the NMCP and MSEPA, looking at importation using a strategy very similar to the one used in polio, examining sequences from Richard Toll and then comparing them countrywide. And similarly there, there's evidence for year-over-year persistence, indicating there's probably both importation and local transmission. And finally, in the South, and we have less data in the South, but again, interventions were introduced, scaled up in 2013. If we just look at the overall change in multiple genomes, monogenomic, and clonality, you see the interventions led to this change, perhaps indicating even in areas of very high impact, this data will be useful to understanding program progress. And so, but of course, I think we should blue sky HRP2 deletions. HRP2 is the basis of RDT diagnostics, the billion diagnostics used. If the gene is deleted, the parasite's not going to be detected. This is something that we could do today, and I think is something that, again, some countries are doing, but it's all, again, in this research area. Could this become part of the standard surveillance so countries will know how accurate their RDTs are? If we go further, perhaps genetics actually would allow us to understand, to be able to predict vectorial capacity, particularly in areas that are eliminating, where the question is, can an imported parasite actually be transmitted? There's work from Carolina Barillas Murray's lab looking at PFS47 and its polymorphisms associated with different geographic regions and associated with different abilities of mosquitoes to actually become infectious with this or become infected with this parasite. Similarly, the genome projects, many of them now, looking at the potential for being able to predict vectorial capacity. These are still very much research projects, but I think by combining the needs of national malaria control programs and the research community's kind of innovation, we may very well be able to fast track some of these things, and I think it's worth thinking about. But let me finish by saying, I sort of feel, and I put this up as a tug of war, kind of as it's not really, it's sort of an intellectual tension between those who would say, let's just continue what we're doing. We know we can eliminate malaria. We have in many places without any new tools. Why should we take the National Malaria Control Program's precious time and introduce new technologies? I think it's important to have this intellectual question, and it's important to ask critical questions for the malaria elimination. But maybe a better way to think about this is maybe DNA could actually bridge the gap between the current surveillance and what's actually going on in communities because of this underlying principle that I showed you in terms of being able to get the granularity of what's actually going on in these infections as it relates to transmission. So with that, I would say I'm strongly in favor of the idea that we introduce this form into malaria surveillance. And in fact, I will go so far with my colleague in the second row to say that perhaps we should have a new logo in the global malaria program that includes DNA as part of the tools that we need. And let me finish by just reminding us of Al McGill's focus on evolution and competition. The Red Queen hypothesis is what he always talked about, and he was a wonderful teacher. Here he is teaching in the science of eradication, sharing his knowledge with those mid-career professionals, those National Malaria Control Program individuals who come to the science of eradication to learn about the latest ideas. With that, thank you.